Okay, welcome everybody to the Miami Project Wednesday seminar series. Today it's my pleasure to introduce Dr. Ashutosh Agarwal, Ashu, who many of you probably already know. He's been a faculty member in the biomedical engineering department since 2014. And is also a co-director of the Bionium, the Biomedical Nanotechnology Institute at the University of Miami. Just to give a little background on Ashu, so he graduated with his engineering degree, bachelor's in engineering from the Indian Institute of Technology, IIT, which again, all of you might know is one of the premier institutes in India. Then he went to University of Florida for his PhD in Master's Material Science and Engineering. After that, he was in Columbia and Harvard at the Wyss Institute doing his postdoctoral studies. That's really where, he started and developed this human diseases on chip model from working on cardiac tissue on chip model first. Since then, it was highly recruited at a lot of places, and we were lucky that at the time Bionium had and Dr. Cody, Richard Cody, who was the chief of pathology, the chair of pathology at the time, and BME could hire him jointly at the University of Miami, and he came in working actually, I think he came in and the first year he came in, they got a U01 grant with Sherry Stabler at Diabetes Research Institute to develop an eyelid on a chip, like a pancreas on a chip model. And since then, his career has really taken off. I know last year he published somewhere between 8 or 10 papers in very high impact journals in this area. He's been working on several multidisciplinary U and R grants, developing these organs on chip models, and certainly is of interest in terms of neural engineering, as well as we start thinking about how do we develop brain on chip and sort of neuronal tissue on chip models, right? And so Dalton and I wanted him to come in and speak to the Miami Project faculty today about the work that he's been doing. And so with that, I'll turn it over to Ashu.
Well, thanks, Shira. That was one of the nicest intros I've gotten anywhere. And that just speaks to how nice you are yourself. So I appreciate it. Before I get into science, because I'm talking to Miami Project, I thought I could include the initiative that's really been my, the closest to my heart over the last summer, 2020 summer, coming into the spring. So this was written over the end of summer, starting fall, when, with the horrific murders of George Floyd and killing of Breonna Taylor, we had university do a deep soul search on how they can contribute towards racial equity on campus, not just through academic studies, but through concrete steps. So I led a team which got $100,000 funding from ULink that, obviously John was one of the founders of the ULink initiative. So I appreciate what he set up, and I saw he was on the call, so I wanted to give him a shout out. I served on the ULink team as an action team member in the first 2 years, and then I ended up switching out, and we are leading, along with Katie from Miami Project, a team of PIs from medical school, biochemistry, psychology, neuroscience, Miami Project, biomedical engineering. In fact, all engineerings are represented, and a big chunk of that $100,000 will go towards is going towards actually 11 Black undergraduate fellows that we've recruited, who would not have worked in a research lab but for this funding. They were all doing on-campus jobs, pretty heavy on-campus jobs, and so we wanted them to not have to make the choice between doing, sort of very mundane on-campus jobs and working in an immersive lab on campus. And we chose very carefully the host labs that could host those 11 undergraduates. And then during the spring, as we started coming back in person, they have, launched their research programs, their own little research programs. We've connected them to various on-campus professional networking opportunities led by NSB, National Society of Black Engineers. So NSB ended up selecting me as their faculty mentor. So that really helped enable, the integration of my program, which I call JANUS, Joint Academic Nurtureship for Underrepresented Students. You have to come up with an acronym, obviously. And JANUS program is now well integrated with the NESB program. Sort of as a payback or return on our investment for the stipend that these students are receiving, and they're all getting paid at a healthy $15 an hour. They have to do one-to-one mentorship of high school students. And these high school students come from two established programs at one run by Dr. Wendy Cavendish in Education and Human Development. And she's been running an Inspire You program that was initially funded by Donna Shalala. And then, she's been sort of running it on, kind of keeping it alive. And that is a one-to-one peer mentored program to Black high schoolers who go to Booker T. Washington High School, which is like just in our vicinity at the medical campus. And so Wendy has identified 4 high schoolers who now have an undergraduate mentor assigned to them to demystify careers in STEM, to demystify college in general. 7 of those high schoolers have come from the program run by Dr. Keely Stewart in law school, which is the First Star Academy. And the First Star Academy has students, Black high school students who are in the Miami-Dade foster care system. So real at-risk young people who also got assigned, undergraduate fellows as a one-to-one mentor to kind of talk about, higher education and things like that. So those are the 2 main thrusts. And then I'm also getting help from Dr. Dutheli and Lindsay Thomas from English department to develop digital humanities modules, wellness modules like yoga, breathing, just meditation, mental health, to make sure that these 11 undergrads and the 11 high schoolers all also go through those modules. So these are like the names of the 11 undergraduates, and then you can see their majors are from biomedical engineering, neuroscience, psychology, architecture, mechanical engineering, kinesiology even. And 3 out of those 11, with help from Katie, are actually at Miami Project. And one of them is actually directly working with Katie. So Dionysia is actually working directly with Katie. So I really appreciate the help I've gotten from Katie in launching this program. And these are just their little headshots, but I also wanted to point out that we have done the assignment of the high school mentees. So these are the high school mentees, and as I indicated, most of them have come from this First Star Academy, which is an amazing program not many people know about, run by the law school, and some have come from the Inspire You program. So we're very happy. Next week, we're doing mentor training and onboarding. So we make sure that these mentors know what they're getting into, what the expectations are, what are some of the boundary conditions that they have to adhere to while talking to these minors. And so things of that sort. So like things I'd never done before, but I really got into it over this summer. I don't know if Katie's actually on the call, Shurud, is she? But she wanted to say something.
I don't see her.
Okay, all right, that's cool. Yep. All right, so with that, let me just switch quickly into my, the day-to-day of what my lab's been doing since 2014. So as Shurud indicated, my lab works on designing what we like to call microphysiological systems, organs on chips. NIH calls them tissue chips. And the problem that NIH recognized way back in 2010, when I was just starting my postdoc, was the declining productivity of discovering new therapies. So on the y-axis here, you've got number of drugs that a pharmaceutical company would be able to bring to market with a $1 billion investment in R&D budgets. And the y-axis is on log scale. So you see a complete exponential decline in the number of drugs that could come to market, and this is adjusted for inflation. And we're well above an investment of $2 billion per new therapy. And the idea is that these therapies, the bulk of the cost and time for development happens in clinical trials, and you really don't want to fail at the human clinical trial stage. And so to improve the chances of human clinical trial success, you want to make sure that the models on which these compounds are being tested are replicative of human outcomes. And the way to perhaps do it is to combine the throughputness of cell culture systems and the whole organism responses that animals provide, but do it in a human relevant fashion. And perhaps not just do it for one organ system, but do it for multiple organ systems. So if a drug had organ-organ interaction going on, you could couple these organs at will and really reveal drug interactions and a now that was the original pitch. And so this was the concept figure we drew in a DARPA grant when I was a postdoc, which ended up getting awarded to the tune of $35 million over 5 years, so $7 million a year. And the idea was to design 10 living human organs, connect them microfluidically together, have them live for 4 weeks, and do a drug response curves and connect drug response testing, and compare that data to clinical data. But it, and over the last 10 years, I think the application has sort of moved away from doing drug discovery to doing more disease modeling. As iPSCs have become more prevalent, as access to genetically modified cells is becoming easier and easier, I think the sweet spot for organs on chips perhaps does not lie so much in doing, toxicity assays or efficacy assays. It lies perhaps in replicating rare genetic disorders on a dish and then delving deeper into mechanisms of those diseases. So when I started at 2014, here at I looked around myself, what are the areas of key strength? And my previous program officer from NCATS told me that they were gonna, come out with an RFA on doing eyelids on a chip. And I never done diabetes research. So I went and had meetings with the leadership here at DRI, who were always, anyone I've always contacted at has been very, generous and gracious with their time. So I've appreciated that here at Miller School a lot. So obviously, pancreatic islets are being explored for clinical islet transplantation for type 1 diabetics. The problem is that the islets that come out of a human cadaver have a very short shelf life. And NIDDK solicited applications that would improve the shelf life of these islets, and perhaps you could also do functional testing on these islets before they go into a type 1 diabetic, or you could even predict the kind of immune responses that this type 1 diabetic would have post-transplantation, so that you could manage this person who's already an autoimmune patient in a more personalized fashion. Now, let me just back up and introduce a bit of fluid physics here. Normal microfluidics have very small volumes and very low clearances. So maintaining a predictable flow within these devices is often not an issue. However, when you're switching to these very large 3D spheroids, organoids, vasculoids, and by very large, I mean anywhere from 100 microns to even 750 microns or so. One really needs very large clearances, very high clearances in the device, so you could have these spheroids, organoids living in the cavity. Now, when you start increasing the height of the channel, you start losing some of the predictability in the flow patterns. I'm simplifying the language a little bit, but the idea is that having large clearance devices makes a microfluidic flow profile very problematic. And that's sort of the secret sauce behind the devices I've designed here, is to still be able to create sort of a non-turbulent flow in these devices. And the way we do it is through these geometrical tricks, such as having chamfer cuts at the entrance or obstacles at the entrance, so that you could really direct, let's say you were coming in with different glucose concentrations, that glucose concentration really goes down to where your biology is. And then to avoid any dead spots, dead zones, you also do some tricks like you narrow out the outlet, so you kind of force the liquid out and speed it up. So it's not laminar, but it's also not turbulent. And you can solve for, the good thing is you can solve for the effects of all of these geometrical perturbations in the device geometry in a computer model first. In the same computer model, you can input parameters for, let's say if it's an islet, for insulin release as it relates to local glucose concentration and local oxygen concentration. So if you switch from a low glucose to a high glucose, you can kind of predict, in a computer model, what the insulin release would be. And then you can add in through established mechanism and relationships, how that insulin release would depend on local oxygen concentration. So I relied heavily on the groundwork, on the simulation groundwork laid by Peter Buchwald, also at DRI, and then we ended up publishing a combination of these things on the cover of Lab on a Chip, again, a couple of years ago. And that really established the design, build, test philosophy that I've followed for all projects in my lab. We do all those 2D console-based flow simulations. Once we're happy with the geometry, we take those 2D geometries and convert them into 3D computer-aided design. We take those 3D designs and then do all the fabrication prototyping in-house in like these inert plastics, like the plastics that we all already use for cell culture. So no, exotic materials. And these soft plastics are either milled or cut through laser cutters, or we even do 3D printing now. And the devices are always fabricated in these two plastic pieces that close onto each other like a Lego. So two interlocking Lego pieces that are closed and held shut by a mechanical clamp that we also got custom fabricated through a company in the Netherlands. And this clamp is just like, it's a mechanical clamp, and it closes these two pieces of plastic together. Between these two plastic pieces, we run a custom made rubber gasket, which is shown in green here. So you've got 3 channels here. You've got 3 wells for manually placing whatever biology you want. And then the rubber gaskets get compressed by the force of application of the clamp. And now you get fluidic seal. Now you can run 3 independent experiments. So that's like one of the simplest devices. And this simple device actually works really well, both in our hands in Miami, but in hands of collaborators in Gainesville. You got rat islets, mouse islets, human islets. And if you look closely on the human experiment, the classic experiment for insulin release is you let the islets sit under low glucose. So you have them in a fasting state. And then as soon as like right after lunch, you should have a high insulin spike. And then you should have a plateau insulin at a lower level to hold you till you get hungry again, and then it's time to eat dinner. And so that's the kind of response you want to see. And we do see that on human islets. But you also see that on the same islets tested within the device, kept alive within the device over a week, a complete decline in insulin release. And this is the problem that NIDDK wanted us to, kind of combat. So to do that, and this paper just got published in Science Advances, again, a collaborative paper between me and Dr. Stabler, who's the other PI on the grant, is to not just have a device that's composed of an oxygen impermeable inert plastic, but have features in the device. So the floor of the device is a through cut. And at the place of the through cut, we have adhered an oxygen permeable membrane. And doing these plastic adhesions in a sterile fashion that could withstand 37 Celsius over long periods of time was just like this complete deep dive into material science. And that's sort of my comfort zone anyway. So we are very happy now that we have a suite of membranes, and the technical name is perfluoric alkoxy membrane. And these membranes, based on their height and oxygen diffusion properties, we understand how these membranes would let oxygen through them independently of any fluid that you would bring in through the regular channels. Within the same device, we fitted oxygen spot sensors for in situ oxygen measurement that are read off by fiber optic cables. And you can kind of see those black fiber optic cables. So now you got a device which allows you to control local oxygenation, local perfusion, and measurement of insulin through like ELISAs on the outflow, but local measurements of oxygen consumption as well. And as I said, it's always important for me that the device works both in my hands and in the hands of biologists. And so it's very easy to use. You can cast your gels, you can cast your biology manually, you can close the chip when you're ready for fluidic interrogation. You can put the chip in an incubator and have it live for weeks on end. Or once you're ready for functional assays, you can bring the chip out and hook it up to a fluid handling system or a microscope. And you have all the assays available to you. And having an oxygen permeable, independent supply of oxygen permeable feature really helps. So after 7 days, you can see that the islets, which had lost their function, actually do retain their insulin function. So insulin's on the y-axis. And that's unsurprisingly revealed also through live dead staining. That was rodent islets on the previous slide. This is the money slide with human islets. And so these are primary human pancreatic islets, right? So at 72 hours, again, we see good retention of function. And this project has really blown open a lot of collaborations and hopefully potential funding opportunities in the future. Dr. Stabler's lab, who used to be in Miami, who recruited me to Miami and then promptly moved to Gainesville, her lab's expertise is in assessing the biomaterials that can prevent an immune attack, that can hide these islets from an immune attack. In another collaboration that I've been developing with Doug Melton, who's the director of the Harvard Stem Cell Institute, you might have seen these news reports come out. His company, so he published a cell paper for the first time he could derive beta cells from embryonic stem cell sources. And the company that he founded in Cambridge was just bought by Vertex for $950 million cash. And they just announced approval for a phase 1/2 clinical trial for transplantation of beta cells that have been derived from human embryonic stem cell sources. So it's very exciting days for type 1 diabetes, cures. But these stem cell derived beta cell clusters have to be tested for function. And, and so we're hoping that we can apply our devices towards doing that functional testing in association with Dr. Doug Melton. The third application is in, as I said, in doing disease mechanistic studies. So here is a movie, again, done in collaboration with stem cell researchers at University of Florida, where you have a beta cell cluster in red over here, and derived from iPSC sources. And from the same human, from the blood sample, they have derived T cells, which I'm recirculating in the chip. And as you can see, the chip is amenable for good like imaging, time-lapse imaging. But at some point, you do see the T cells come and home towards that islet cluster and then destroy it, right? So these kinds of very, I think this is very dramatic, but something like this is also happening in the pancreas of these patients. And so I think of these chips as a living histology slice into a person's organ, and that's very powerful. So that's all I had on the diabetes story, and then I can switch to the other funded work in the lab, and then perhaps some unfunded work. But if I could summarize the diabetes story is, the design build test paradigm is pretty well established now. I'm always looking for partnerships to companies that can supply the associated hardware. So Micronet is the company in Netherlands that makes those clamps, and you can buy that clamp directly from them. We also have invested a lot of time in coming up with fluid handling systems to operate the chip. And we designed the architecture of a fluid handling system that you can actually buy off the shelf from a company in France called Fluigent. They call it the Miami box. And I'll talk more about the fluid handling capabilities towards the end of my talk. I myself have spun out a company called BioVitro, mostly to service the growing number of academic collaborators, where, so all of these places, WashU Nephrology, Hebrew University in Jerusalem, and the Diabetes Institute, UPenn Med School, and Indiana School of Medicine, and Vandy School of Medicine, all have my chips living there. And so to service those collaborations was becoming untenable to put graduate students on it. So we really hope to be able to do it through a commercial entity, and we call that company BioVitro. Oh, I'm sorry, one last story. So this paper came out last year in JCI, where researchers at Vandy School of Medicine, Al Powers and Marcela Borisova, take human islets, dissociate them, and then they can transfect either alpha cells or beta cells with certain ion channels, light activated ion channels, and they can close and open these ion channels based on application of light. So they really needed a device where you could apply light stimulation at different wavelengths, but then also measure the hormone production through a perifusion assay. So the chip was a perfect application. It was a good combination of what they needed and what my lab could supply. And we generated a lot of cool data, which, as I said, this JCI paper has 3 corresponding authors, 2 from Vandy and 1 in my lab, who provided the engineering platform to do all of these beautiful assays, where you're turning light on and off, and you're either stimulating hormones secretion or you're suppressing hormone secretion. You can even transfect cells with a GCAM calcium reporter, and you can measure both calcium production calcium signaling by these cells and hormone production, all in this platform. Very simple platform that allows imaging and perfusion at the same time, concomitantly. So that was a lot. I thought we could stop here and see if like anyone had any burning questions on the organoid story so far, and then I'll quickly talk about cancer.
Sure. Any questions from the audience?
Ashu, that's so far very interesting. Have you looked at anything along the lines of stem cell differentiation, utilizing your organoid platforms?
Oh, so you know, my classic training during my postdoc was in cardiac tissue engineering, and not so much for differentiation, Dr. Dietrich, but for maturation of newly differentiated stem cells. We want to apply like this. These chips allow a lot of bioengineering stimulation, stimulatory environment that might serve to further mature what's often a neonatal phenotype of a newly differentiated stem cell, specifically cardiac cells. So, you know, well-recognized problem is the human iPS differentiation leads to sort of a neonatal state or an atrial myocyte, not a ventricular myocyte, that's real strong myocyte. And we think that during early development, the exposure to higher fluid pressures, stretch, and those kinds of things forces the cells to lay down more sarcomeres and become more strong. And that's what I've been focusing on maturing cells. Now, this philosophy can be applied to stem cell derived beta cells also. So one of the grants that Sherry and I wrote to NSF was to keep applying, like sort of like keep exercising these cells, keep exposing them to low glucose, high glucose, low glucose, high glucose, and make them work. And I called it gym on a chip, but like kind of force them to, you know, do the work, and maybe that matures their phenotype, but not so much towards differentiation. I just don't have the technical background to like actually tackle the differentiation problem. My newest graduate student, didn't include the story, is Dan Pelez from Boston Palmer, and he has an R01 on growing retinal organoids. And it's so interesting that the self-organization and then the self-differentiation of a retinal organoid is driven by the oxygen, like the atmospheric oxygen that the front experiences and the lack of oxygen that the back experiences. And it's like a 30-week long differentiation protocol. So what he needs to do is induce a stable oxygen gradient in his retinal organoids to perhaps drive the differentiation of the retinal organoids, and then also perfuse them to keep them supplied with nutrients and whatnot. So that would be our first foray into creating a bioreactor, essentially, to differentiate these really huge retinal organoids. So he and I are co-funding a PhD student who just started last fall. Okay, so let me go back to sharing. So the next funded study in the lab is on, again, a collaborative effort that established a liquid biopsy-based consortium for early detection of breast and prostate cancer here at the Sylvester. So it's led by me and Dr. Richard Cote, who Shrud mentioned, was the ex-chair of pathology, who's now moved to WashU. It's sort of the story of my life. All my collaborators end up moving. And he had pioneered a microfilter technology that does a size-based separation of cancer cells in the bloodstream of cancer patients and, you know, large white blood cells, but lets all red blood cells fall through this microfilter. So you can very simply just run blood through this microfilter surface. And at the end of that run, you can recover the microfilter slide and then stain and count the number of cancer cells. And the number of cancer cells will give you somewhat of an idea of the metastatic state of that patient. Now, this microfilter technology was well established before I even joined, and I've done some engineering contributions towards making that technology robust, making a box around doing all the fluid handling. That was quite easy and trivial, but it really helped with shipping these boxes at different sites that were running, you know, sort of clinical trials to quantify the number of cancer cells. And again, this is not my work, but Dr. Cote has shown that because it's a purely size-based separation, not an antigen-based separation, you can do all kinds of solid tumors and even non-solid tumors you can recover circulating tumor cells from all kinds of cancers. What he and I have been doing over the last 2 years, so we are in year 3 of this clinical trial-based study, is collect blood from stage 1, 2, 3 breast cancer patients, 5 tubes per draw. 2 tubes are meant for quantification of circulating tumor cells and associated cells of the cancer microenvironment, such as circulating cancer-associated fibroblasts, MDSCs, clusters of those cells, what have you. 2 tubes are biobank right now for doing circulating cell-free DNA analysis and quantification, and 1 tube is biobank for doing all kinds of extracellular vesicles, such as exosomes, etc. And the idea is that in addition, or instead of just doing quantification of cells or clusters of cells, all of these things will also contain and add some sensitivity to early detection of breast cancer. And not just early detection of breast cancer, but also if the patient is sort of already cured, a risk of recurrence, or managing that patient better. And so if you can collapse the readouts from these very different assays into a singular metric by assigning weights, which are unknown right now, by assigning weights to each of these readouts into a single metric to manage the patient better. So we have recruited, we've promised to recruit 400 patients in stage 2 and 3 and 200 patients in stage 1, and have sequential, so both in the neoadjuvant arm and in the adjuvant arm have sequential collections. And because Dr. Cody moved to WashU, the grant was split, and that actually helped us because now we added another site for recruitment of patients. So we're recruiting both at the St. Louis site and at the Miami site. And my other contribution to this grant is to recover these circulating tumor cells. So my first research paper at Miami was in collaboration with Dr. Cody, where I invented a technique to not just capture these circulating tumor cells, but release and recover these circulating tumor cells in a viable fashion. And we do this through this this coating that changes its surface properties based on whether it's at room temperature or it's at 37 degrees Celsius. And once you release the circulating tumor cells, you can play with these circulating tumor cells. And one of the things that we wanted to do was help understand the changes that happens in these cells when they decide to extravasate, let's say to a bone or a lung or a kidney, and post-extravasation when they decide to proliferate and create micrometastases. And so this was led by my graduate student Vera Mayo, who just graduated, who we dearly miss. And she kind of designed a crazy looking chip. And the chip she designed was a chamber for keeping the circulating tumor cells that have come from patients in circulation on a bottom channel, which is in yellow. At some point above the yellow channel, there is a membrane, and the membrane is populated with cells and perivascular cells. So these are the pericytes that literally pull the cancer cells out of circulation and bring it to a bone compartment. And the theory that we're chasing is that it's the critical role of these perivascular cells to kind of make these cells extravasate and cause all kinds of problems. So we've put these cancer cells in association with just the perivascular space and both with the perivascular space on the bone. And this paper was just published in 2021, and we did a deep dive on the kind of molecular changes that these cells undergo when they're in combination with just perivascular cells or in combination a sequential exposure to a perivascular environment and a bone microenvironment. And it's very interesting. And in future, my current R01s to NCI have focused on going deep through this device, through access to all of these patient cells, going deeper into disease mechanisms of extravasation and proliferation at the micrometastasis site. In future, perhaps if we could just divide the circulating tumor cells, we don't have to cut into the patient to get a biopsy to do drug sensitivity, chemosensitivity assays. If we can divide the circulating tumor cells, we can create a biospecimen from these patients to kind of test what kind of chemo cocktails that she needs to be on and do drug discovery in a personalized fashion. So yeah, aim 1, aim 2, aim 3 of my R01s, which haven't been funded as yet. I'll skip over the work with Google Research, but I'll get to a third generation of the device. Whenever I show my chips to potential collaborators, they all want to know if they can transition a transfer culture onto a fluidic platform. So many labs at Med Campus, are very happy with doing transfer cultures where you create a physiological barrier system for modeling any kind of barrier. So these barriers could be air-liquid interface barriers in the lung. These barriers could be glomerulus barriers in the kidney. And what we have ended up inventing is a way of, so I'll skip the physics. Of course, we understand the physics of our system. What we've ended up doing is inventing a way of having your conventional transfer cultures live, attain maturity. So, they form a tight barrier. And then you can manually cut out this transfer with living bilayer cultures and stick it in a chip, in a wet adhesion in a sterile manner, all of which were not trivial from an engineering point of view. And once they're in the chip, you can do all kinds of things, right? You can expose the two sides to different pressures. You can dry out one side and induce an air-liquid interface. You can expose the two sides to different kind of shear forces, or you can basically design your experiments for doing different stimulations on either side. Now that becomes relevant for, let's say, the lung. So in the lung, you have air-liquid interfaces, but you also have the lung stretching during breathing. So we took primary lung microvascular endothelial cells and primary alveolar epithelial cells, created an air-liquid interface on a transferred that air-liquid interface culture to a chip, exposed the top of the chip to different pressures. So the membrane was actually flexing down. And so we created that classic lung on a chip experiment in a modular chip. And then the second case study in collaboration with Dr. Alessia Fornoni in nephrology was to take podocytes and glomerular microvascular endothelial cells and create an organ-specific barrier and expose the two sides, the urine side and the blood side, to different physiological fluid pressures, and have that be the driver for, let's say, filtration of albumin from one side to the other, right? The other engineering readouts you can do on this chip is like kind of measure what's called a trans-epithelial or a trans-endothelial electrical resistance. So the idea is that you're holding the two sides on opposites, you're holding two electrodes on opposite sides of the membrane, and you're measuring how much current leaks through it based on the voltage you've applied. And if the cells have formed a nice barrier, there would be less current, and so the resistance would be higher. So you see cell cultures, go up in resistance and then plateau. And when they've plateaued, that they've attained as good a membrane as they ever will. So yeah, so this barrier function chip is finding a lot of applications and hopefully funded studies. The only funding we had on this project was an F31 application that my pre-doc student received, and he had the benefit of Dr. Alessia Fornoni serving as a co-mentor for his work on podocytes on a chip. So I see there was a question here. Sorry, I just, okay. All right, I thought we could stop here again, Shurud, make sure people were awake and take questions on anything so far. If not, we can go on.
This is Dalton again. Can you look at tumor migration?
So primary tumor migration?
Yeah, we have a neurosurgeon and that's looking on brain tumor, and his whole thesis has to do with the migration of the tumor cells. So I'm just wondering if these platforms could actually look at migratory properties of a cell, for example.
Nice. Yeah, certainly. I'd be happy to have a conversation. Certainly. Everything you've described is within like the laws of physics that the chips live with.
Okay. Yeah. Imagination, right.
Yeah.
So I have a question.
Yes.
Sorry, go ahead.
Okay. In terms of the first part where you said the size separation, you're just relying on literally a screening technique. Have you thought of relying on like the surface charges or even simple inertial behavior. Because you have a flow system, we could use just inertial properties to size classify. And if you incorporate surface charge effects, maybe you get another parameter separation.
100%. Dr. Biswas, there's so much work that's happened in circulating tumor cells, such a crowded field. But what you described, both the things that you described have been explored. There are actually companies who have products out there that either rely on the difference in the inertial gravity of cancer cells versus other cell types. So they would create, fluidic flow profiles and kind of concentrate cells based on their gravity and the surface charges. They even have dielectrophoresis chips that would dielectrophoresis cells in a different compartment.
Which might...
Yeah, the danger with circulating tumor cells is to remain novel. And there's hardly anything that's novel anymore in circulating tumor cells.
Right. The second question I had, which is general and probably naive, in a real system, I have a whole colony of cells. So these fluidic microsystems, is an individual cell behavior different from what a behavior would be in a system? The field of systems biology I'll throw in. So what is the difference? Do people study the effect of scale on these things?
Yes, 100%. I mean, you've asked a very philosophical question. People who are much smarter than me do those kinds of work, but you're absolutely right. But I'll say in relation to cancer, that problem even becomes of more significance, right? They have heterogeneity, let's say within a tumor, and there would be a fraction of cells that respond really well to chemo. And you would think that your tumor has shrunk and you're on your way to recovery. But then perhaps the cells, and then the question becomes, what are those cells that hid from the chemo? How have they mutated? So over time, that heterogeneity changes as well, which has significant implications for patient management. Now, scaling, going from, so there is heterogeneity at the single, cell to cell level. When they start coming together to form a tissue or in health or in disease, those questions become even more pertinent. Yeah.
Good. Thanks.
Yeah.
Hassan here. So I want to build a bit on Dalton's question. So there are two main pains in studying invasion of cancer cells into tissue. Now that the whole 3D growth of organoids is worked out, one is how do you image and quantify the invasion? And that's something for microscopy and automation to figure out. And the other pain is because we grow these things, in order for them to become three-dimensional, they cannot be attached. And then so there are these few less than millimeter something diameter organoids. And when you put them in the well, trying to find them is a big problem. Do you think you could use microfluidics such that you can have these docks within a chip that you put in a number of organoids and the fluidics would just orient them so that in each location you'd have an organoid and then you can separate them fluidically and then do the testing? I think if you could figure that out, that could be tremendous for this.
Yeah. So Hassan, we've done that in relation to eyelids. We created simple fluidic traps based on what Dr. Biswas was also describing. And those traps would hold smaller islets in one location and larger islets in a different location. And then you could ask, different questions from, let's say, a smaller organoid than you would from a larger organoid. I will say in general, organoids suffer from lack of assays, right? All the assays that are available in the catalog of our usual suppliers were all built for 2D based monolayer kind of classic cultures. And the 3D offers a lot of benefits, but the biggest limitation in 3D is you're limited in the kind of readouts you can do. So, in my future work, I also want to develop embedded sensing for within the chips to kind of make readouts possible for 3D based assays, right?
Right. I know Vertex has figured out the whole separation based on size a few years ago, and they were using it to isolate blasts from patient blood. But this would be taking it a step further. You direct them to specific locations and then you keep them there and then you separate them volumetrically so you could do separate tests like screening. So it's an additional design feature that could be, something worth starting to think about.
Right. Hassan, whenever you talk to an engineer, they would never say no to anything you want to get done. It's just the matter of having everything's possible. You can build anything you want, really. It's just a matter of having, commitment and bandwidth on both sides.
Sure, sure.
Take the project somewhere.
Let me just talking to Zain.
To whom?
Zain Zaire.
Oh yeah.
I can set up the call. Yeah.
Right.
Okay, great.
All right. So thankfully, I do have something neuronal to share with you. This is unfunded work as yet, but Dr. Mario Saporta, who's a neurologist here, who many of you might know, and I have been working very closely together over the last 4 years and submitting R1s to raise money on a patient-specific Charcot-Marie Tooth disease on a chip. So he specializes in running clinics and isolating cells from patients who present with this rare genetic neuromuscular junction disorder called Charcot-Marie Tooth disease. And his lab provides us with neurospheres which extend out axons along a direction that we direct that axonal growth in. So the axonal growth is always in the north-south direction. And then we derive skeletal muscle cells, like in current efforts right now, from the same iPSC sources. So it's a patient-specific neuronal muscle cell culture where you have distinct compartments for growing, directing axons towards a muscle. So you can start stimulating the neuron and ask the question, what is the conduction velocity and other things down an axon, and what kind of muscle twitching happens in response to a neuronal activation. And all of those will be very revealing for studying this rare genetic disorder. So I'm very excited. This is my favorite project in the lab right now. Now to get there, it sounds very simple on a schematic. To get there, it took us a long time to figure two things out first before we could do any of this. The first thing we needed to figure out was basically a culture substrate on which both neurons, primary neurons, and skeletal muscles can be kept alive and happy. It turns out just the surface chemistry for keeping these two surface cell types active and healthy was not trivial, and we had to go down a rabbit hole of searching for different materials and different surface topographies. So this is 2-3 years of work in one kind of figure. So here you're seeing a base material of a gelatin hydrogel. A gelatin is basically denatured collagen, and at the very top layer of gelatin, we've bioconjugated a layer of laminin that neurons like. And then the top surface of this gelatin laminin hybrid substrate is also micromolded. And so this micromolded gives it kind of these channels in which you can direct axonal growth, and you can also direct skeletal muscle orientation. The other thing we had to figure out was what kind of media would, just like the culture media, would be necessary to, again, keep these two very finicky, very high maintenance cell types happy and alive. And so that was in a follow-up paper. So also in which we did all kinds of media supplements. So there's so many supplements that neuronal media had, and then the skeletal muscles did not like it. And so we started taking out one supplement at a time and kept asking, are the neurons happy? Are the skeletal muscles happy? And whenever we sing happy, you're applying all of those quantifiable assays to answer, how happy they are. So it's a lot of work. And now we have a good handle of keeping both those cell types on the same substrate in the same media happy. We do those experiments on what I have in my lab, a microelectrode array. So it's essentially measuring extracellular field potentials. And we're pretty confident in being able to stimulate the neurons and then measure the neuronal activity in this burst and spike fashion from these neurospheres. And this is a stitched, it's perhaps a zoomed out image. It's not a stitched image of this neurosphere extending axons down these channels and connecting to skeletal muscles in the green, right? And again, we like numbers, so we like quantifying everything as an engineering lab would. So we had to come up with metrics to basically say, how is this image different from this image? So, right, we just came up with metrics such as axonal coverage, axonal length. We have a lot more in those papers. And here you're seeing some zoom in images of that co-culture environment. When the axons do reach the skeletal muscle, they start laying down and clustering acetylcholine receptor clusters at the ending of a nerve fiber. So green is skeletal muscle, red is the acetylcholine receptor. And you see kind of that clustering happening. Not many people have shown an in vitro assembly of acetylcholine receptors. And then right adjacent to an acetylcholine receptor clustering in red, in white you can see and stain for synaptic vessels as well. And I would just draw your attention to this very last bar graph where you can measure the size of this acetylcholine cluster receptor size for a chip that's constructed from healthy cells and a chip that's constructed from diseased cells, and you see a significant reduction in the size. And the N is pretty high, so we have a very good confidence that we are seeing some functional deficits from having disease cells versus healthy cells on this chip. So we're taking the study further with new graduate student. I kind of graduated my first batch, and now I have young blood in the lab to work on these problems. The other problems I think about and potentially write concept papers to DoD is, if you really are serious about coupling these 10 organs together, we from an engineering standpoint, we need to make sure that our organ linkages, our fluidic linkages are robust, right? The chips don't fail because of engineering problems such as bubbles, leaks, failures, miniaturized pumps, valves. And this is, again, not trivial. From a scientific point of view, I'm not sure how we would couple each organ chip versus other organ chips, right? In our body, there is a physiological parametric scaling of a brain to a lung, to a heart, to a kidney. And it's not clear what those scaling laws are and how they transfer to an in vitro system. And the last sort of challenge is, if I do get a response from an organ chip, how does that relate and scale to an in vivo response from a full human being? So very interesting problems from a scientific point of view, from practical translational point of view. So I can place these chips in the hands of users and they're as comfortable with using them as we are with, let's say, 96 well plates. I try and think of it in this three-axis thought experiment, where the cost is on one axis, the throughput of readout, throughput of manufacturing is on one axis, and then the level of sophistication is on the z-axis. And the 96 well plates are very cheap. They have super high throughput, but they're very unsophisticated. My organs on chips are right now super sophisticated, but extremely poor in both throughput of manufacture, throughput of readout, and in cost terms. So I do think there is value in decreasing the sophistication and improving the cost and throughput of these systems so we can translating them in the hands of intended users, which are biology labs, disease pathology labs, developmental biology labs, and pharma. This is a shout out to the talent we have in biomedical engineering here. So this is a senior design project, and I challenged them with coming up with an integrated fluid handling system to operate the chip. So even though we can make a chip that's simple, to actually operate the chip, you need this entire ecosystem of fluid handling platforms. And so they ended up creating this like little widget which can switch between different sources. Oops, let me just forward a bit here. And also move the 96 well plate collection plate. So through a computer graphic user interface, it's switching between a yellow source, yellow source, yellow source, then recirculating the yellow media, then switching out from recirculation, and then switching to a green source and outputting. And this project ended up winning the best senior design project. These are the two companies, and this is the team. All the people in Italics are gone, which sucks, but they're the ones who developed all of the work I showed today. And the people who are not in Italics are the people who have just joined the lab or are going to join the lab. And again, the italized funding sources are indicated, and then the active funding sources are indicated. And again, I can't thank enough at UM, especially at the medical school. So with that, I will stop sharing and take questions. We have only 3 minutes left. Sorry. Thanks.
Okay, Ashu, thank you very much for a very provocative talk. I think the engineering approaches that you described to us today just have so many, as we've heard some of the questions, so many applications to Pacific questions. And also congratulations on the U-Link Award and the Jandis Award. You know, underrepresented students, that's such an important area. So congratulations on everything you're doing in that regard as well. Any questions, please? I did want to say hello to the Dean Biswas. Welcome to Miami. Great to see you, and you're always welcome to participate in the Miami Project seminar. We're very excited about building alliances with engineering, so it's great to have you on board. So look forward to talking to you more about that.
Sure, thank you, and look forward to that. Yeah.
Very good. Questions?
Well, we did take questions during the talk, so maybe.
So you can look at cell mechanisms, right? I mean, you can put a different type of a neuron or an astrocyte or an oligodendrocyte and add certain type of specific toxicity factors and look at mechanisms of injury, those types of things that you could do, as well as regenerative strategies, as you know, adding things to you know neurites and trying to see if you can enhance regeneration. All those things are possible, correct?
Dr. Dietrich, you mean at like sort of the single cell level or at the tissue level?
Single cell level, I think, or
yeah.
tissue
So
level.
the classic applications of microfluidics is in what you're describing, right? At the size scales of single cells or a few cells, and really improving the throughput of testing, like map out a huge parameter space. And I'm happy to assist in those projects, but they've not been the main driver of what my lab does. My lab has really found its sweet spot in this tissue scale and even like large tissue scale, where you give up on the throughput, but you really enable culture and interrogation of larger systems. But I also run the I also help manage the nanofabrication center. And that's where you would create all of the devices that you just mentioned. And I'm happy to service those kinds of work. Perhaps not directly through my research lab, but you know, through clean room.
Understood. Very good. Thank you.
Yeah.
Okay, it's 1 o'clock. If any no other questions, thank you again, and everyone have a fantastic day.